Yes, okay, dear students. Today we are going to discuss about the Pteridophyte in Selaginella. The last class we completed Xylotum and Lycopodium. Okay, but now here we are going to one more important unit. So that is of in Selaginella. Okay. So we are going to discuss about Selaginella. After the Selaginella, we'll go to one more important one. Yes. So first one. Yes. So here now you see this is the classification of in Selaginella. The classification of Selaginella, that is of in Lycophyta and Bryophyta. Order, that is of in Selaginalis. Family Selaginellaceae and Selaginella. Yeah, here I am. This is a Selaginella plant. Okay. So a little bit variation Lycopodium and this one. So this plant is also sporophytic plant. Okay. Yeah. Before going that one, you see the Selaginella. We are looking. It is a almost all worldwide distributions, cosmopolitan distributions. Here it is in. So here we are looking more than 700 species is there. So majority of them occur in tropic and subtropics. Few of the present in where the arid zones, some are cultivated, some member that is some are the ornamental species also is there, Selaginellas. Okay, and some members of the Selaginella when you are looking, it is a xerophytic characters also is there. Okay, some of the xerophytes plant-like structure also we are looking in Selaginella. And when we are looking this plant, it is looking like a ball-like structures. Okay, so when you are looking in the summer, it is looking like a ball-like structures is there. Okay, so this is of an occurrence. Yeah, yes. So now we are going to Selaginella species, but whatever it may be, Selaginella is also a sporophytic plant body. So this sporophytic plant body means differentiated plant. That's all. Root system, stem system, and the leaf system is there. Yes. So when we are looking in Selaginella species, you see here. So that is of in Stricogynandrium and Selaginellaceae. It's of a genus type means. So when we are looking the plant according to structures, similarly, you see the Lycopodium plant. Lycopodium plant also is a genus. We are looking the two types is there. Similarly, here also we are looking two type of species is there. The based on the habitat and nature, the leaves of Selaginella species can be separated in two groups, means subgenera. So one that is of in Stricogynandrium and second that is of in Selaginella. How can we identify it in both genus? So you see now the first one, when you are looking in the first one that is of in Stricogynandrium, leaves in plant are different, hence is called of heterophilous. Leaves and plant are different. Okay, leaves or plants are the different shapes you are looking. So that's why here it is in you are looking like that. Yeah. So and next one that is of dorsiventrally symmetrous and leaves are arranged in rows. The plant prostrate with the short erect branches. So when you're looking, these are all the structures. These are all the structure that is of in Stricogynandrium. Yeah. Similarly, second one that is of in Selaginella, leaves are identical. Hence, that is of homophyllums. So means when you're looking in these type of these subgenera species, all the leaves are similar, same type of the structures is there, same type of the shapes is there. So that's why we call that is often isophilous leaves. homophilous, same type of is there, and that is of dorsiventral, this of an isometric. So here the leaves are arranged spirally, 1, 2, 3, like that, spiral arranged. All these plants are erect. So here this is of two types of genus is there in Selaginella plant. Okay. Yes. So next, now you see here the leaves are spirally arranged on the stem. Roots are adventitious, rising on the underside of the prostrate or creeping stem. Okay, how we are looking these plants? So how can we identify the Selaginella plant? Okay, so the Selaginella, the leaves are arranged. Leaves are arranged spirally is there. All the roots are adventitious roots. So these roots are comes from the underside on the creeping stem, and the creeping stem. The stem we are looking like the creeping structures, all the roots are coming inside the stems. Okay. So the species, some species that is often erect or ascending branches also you are looking. The special structures are present called often rhizophores. Okay, rhizophore. This is the most important structures. So that is what is the meaning of rhizospores? So rhizophores are specialized root-like structures. Specialized root-like structures found in where these some Selaginella plant. Okay, so which belongs to the divisions of same all the so Selaginella plants. Okay. So what is the main functions of in this one of rhizophore? That is of anchorage and support. So and nutrient water absorptions, vegetative propagations. Yeah. So these are all the main functions of rhizophores. Yes. So next, now you see here, this is the plant body, sporophytic plant body. So now you see here, this one, this is of rhizophore. Rhizophore means one of the structures comes to just above the one above the stem, not above the stems, just here. It goes to the underground. We call this often rhizophores. Same, it is also acting as a root like. Okay. So, but one branch is coming from, it goes to directly where the soil. That's why this subspecial structure is there. Yeah, here this is often rhizoids, here it is often stem. But you see, all the leaves are not looking the same. Here, this is one type of leaf is there, and here also one type of leaves is there. Okay, dorsal leaf and ventral leaves is there. And tip of the branches. So we are looking the spikes, means the cone-like structures is there. So entire the structures we call that is in sporophytic plant body. Okay, sporophytic plant body. Yes. So now you see here. This is all the external features of plant body. So external features of plant body, that is sporophytic plant body, and here it is in root system is there, stem and leaves is there. Okay, different type of the leaves are also we are looking in this one, different type of the shapes, different type of the size also. So we are looking Selaginella. This is of external features. Yeah, next now you see we are going to the internal structures of plant body. Internal structures. So first now you see here the rhizophore internal structures. Same here, rhizophore internal structures. First one, that is of in rhizophore means, yeah, already you know very well the rhizophore. Here also three regions is there. One, it is in epidermis. Second, that is of in cortex, and third one, that is of in stele. Third one, that is of in steles. So mainly three regions is this. Okay, one, it is in epidermis. Here now you see. So epidermis, it is a single layer. There is no intercellular spaces, but above the epidermis cells, so we are looking one special wax-like structure is there. Means one layer-like structure is there. We call that as of cuticles. So that's why this of little xerophytic characters is there. Yeah, and below the epidermis, above the stele. So here we are looking cortex, but cortex it is a now you see here this one below the epidermis, one or two or three layers is there. It is in a cortex is there. Yeah, and also now you see here this one. This is also lower cortex. Also you are looking in this one. So it's above the cortex. So here you are looking hypodermis. So what is a hypodermis? Single layer where the thin wall-like structures is there. Thin wall-like structures means little advanced cells also you are looking in that one. So hypodermis, some of the species of hypodermis, it is a thin hard walled is there. We call that is of sclerenchyma. So what is the main function of sclerenchyma? Means here sclerenchyma, it gives to the mechanical support. Okay, that is the main function of sclerenchyma. Yeah, next one, that is of in cortex. So here this is all to the cortex. This cortex is total made up of in parenchyma. So smooth walled structures is there. So these are all the cortex is contain different type of the food materials. So many types of food materials, oils, fats is there, and oils, all type of materials is contained in this ones. Means it gives to the food material entire the plant. So here this stuff in cortex. Okay, yeah, and next one, last one, that is of stele. Here already stele is two type of cells is there. So one, it is in xylem, and the second one, that is in phloem. Okay, xylem and phloems. And here now you see here, yes, so the phloem present around the xylem. This is most important. Phloem is present around the xylems. Some of the species, xylem present a central core with the protoxylem and exarch positions. With protoxylem is at center. Some of the species, the pith is of entirely absent. This is the structure. So now you see here this one, this of in center part of the stele. Okay, so now you see the rhizophore. It is a three regions is there: epidermis, cortex, and stele. That's enough. Yeah, next now you see the leaf section cutting. Here now you see Selaginella leaf section cutting. Here also three regions is there. So one, it is in epidermis. So second, that is of in mesophyll, and the third one, that is in vascular bundles. Okay, the Selaginella epidermis, it is a single layer. There is no any intracellular spaces, and above the epidermis structures, we are looking one cuticle layer-like structures. Similarly, same upper epidermis also is there. So lower epidermis also. So here the lower epidermis is contain some hole-like structures. We call that is in stomata. Stomata is there. And next one, now you see in between of so below the epidermis and upper epidermis and lower epidermis, So here we are looking one mesophyll, a spongy parenchyma palisade tissues is there, smooth walled structures is there. So all these are all the cells are contains this is chloroplast. Okay, chloroplast structures we are looking in this one. And next one, the center part. Center part, it is in a vascular bundles or very simple structures is there. So this is in Selaginella leaf. Okay, Selaginella leaf, here it is three regions is there: epidermis and mesophyll and vascular bundles. Epidermis, it is a two layers is there: upper epidermis and lower epidermis. Single layer, there is no intracellular spaces. Above the epidermis, we are looking at cuticle-like structures is there. Okay, and the lower epidermis, so here we are looking stomatas also is there. In between the structures, we are saying that is a mesophyll. Mesophyll is contain chloroplast. Okay, and last center part of that is of in vascular bundles. So this of in leaves. And next one now you see here is a third one. Third one, that is of in stem section cutting. Yeah, here this is the most important. So the stem section cutting, here it is a three regions is there. Three regions is there. One, it is in epidermis. Second one, that is in and a cortex, and a third one, that is of in center part of the section, that is in stele. Center part, that is in stele is there. Okay, yes. So now, yes. So when we are looking the section of the stem, you see here this one is epidermis. Okay, yes. So now epidermis here also, that is of in a single layer. There is no any intracellular spaces is there, and above the epidermis structures, so here we are looking one cuticle, one layer like structures is there. Okay, so here now you see this one. Second one, what is the second one? Here it is in cortex. Okay, so cortex is made up of different type of the cells is there. So what type of cells is there? One, it is of sclerenchyma cells is there, sclerenchyma. Okay, sclerenchyma cells, and below the sclerenchyma cells, so here we are looking some light thin walled structures is there. Thin walled structure cells also we are looking in this one. So what is the main function of in sclerenchyma? It gives to only mechanical support, and below the sclerenchyma, here it's contain different type of the food materials also we are looking in this one. And next now you see here, so one more, so here we are looking lot of air gaps is there. Okay, lot of air spaces also we are looking, vacuoles like structures also we are looking in this one. And next one, here it is now you see, so in steel, the steel it is in so monostelic. Okay, monostelic conditions, and bistelic or polystelic, different members of Selaginella. So we are looking different type of the steels is there. So what is the meaning of monostelic? Okay, monostelic means only particularly center part of the section, that is of monostelic. And bistelic means one and two portions is there, that is of bistelic is there. Sometimes more than two still also we are looking in that one. So that is in polystelic, means based on the plant structures we are looking different type of the cells. Okay, so whatever it be, there is of in monostel is there. Next now you see here this of in monostelic, for example here, and bistelic here it of in one more example is there. Okay, so this is of in external structures and internal structures of in Selaginella. Yeah, yes. So next now you see here life histories of in Selaginella plant. Okay, yes. So life history means here now you see this time sheet. This is of in Selaginella plant. Okay, this is the structures mainly. You see first one, Selaginella plant. Here it is in sporophytic plant body. Hello, please respond. Shahenge bond. Yes, sir. Our audio is clear. Our audio clear, visible, audible. Is it clear? Hello, respond. Your voice is not clear.
Sir, it is coming.
Yes, okay, now. Yes, so today you see, now we are going to Selaginella plant. Yeah?
Yes, sir.
You know the Selaginella plant, but please tell what is the external structure of Selaginella? Selaginella plant body, it is a sporophytic plant body. Okay? Sporophytic plant body means differentiated plant. Yeah. You see, now you see here, I'm showing the life history of Selaginella. Selaginella plant body, it is of in sporophytic plant body. The sporophytic plant body is produced to the spores. Okay. Yes. Now you see here, this is the spores here. Okay. But how the development of the spores means here, where is the Selaginella plant here? This is often Selaginella plant. Okay. So this Selaginella plant, tip of the Selaginella branch, okay, one of the sections, one of the cell is going to modified as a cones. Okay, as a cones. Cone means here it is in asexual reproductive organs. Okay, the cones, it is in asexual reproductive organs. The sporophyte of Selaginella represented in different types of habitats. Many of the species prefer in grow humid shady habitat and very few are epiphytes, even the xerophytic natures. Okay. Now you see here this one, the cones and Selaginella at the terminal bearing two kinds of sporophylls is there. Sporophylls which contain the spores, which contain the sporangium, that is in sporophylls. Phyll means leaf. Leaf-like structures is there. So this leaf-like structure is contained the sporangiums. Okay. So these spores are two types is there. These sporophylls are two types is there. One it is in megasporophyll and second one that is in microsporophylls. Okay. Yes. So both are similar to the vegetative leaves or maybe, so means you see here, this is in also, here we are looking the leaves, but about tip of the branches we are looking sporophylls. Little variation is there, normal leaves too and sporophyll leaves. Okay. Normal leaves are totally green like structures is there, and sporophyll leaves a little colorless structure also we are looking. That is the variations of in normal leaves and other leaves. Okay. Now you see here, one it is in megasporophyll, second one that is in microsporophylls. So yes, they have the one. So you see here, this is the leaves. These leaves is contain the sporangiums. The sporangiums are contain the spores. You see here, this is of in the sporangiums here. This one, this is enough in spores is theirs. Okay. So here this one, this is of in microspore is there, and second one that is of in megaspore is theirs. So total this is of in microsporophyll, and here it is in megasporophylls. So these are all the structures you are looking, tip of the branches, tip of the plant. So here one structure is going to divide anticlinal, periclinal, metatic divisions. How much of metatic divisions is going on, that much of development you are looking in. So we are looking the development. After the development, so the sporangium we are looking like this. Okay. Yes. So here now you see this one. This of sporangia. Okay. So sporangia, one it is in megasporophylls, and second one that is of in microsporophylls. Okay. Yes, so megasporophylls are produced to megasporangium, and microsporophyll are germinated to microsporangiums. Okay, so after the development, in between the sporophylls, spores is there. Okay, so we are saying there is a spore mother cells. It's going to modified as a megasporophyll. Similarly, second one is also going to modified as a microspores. After the development, the spores comes to outside the plant, and both together formations. You see fertilizations. So we are saying that the fertilizations. After the fertilizations, here formations. One vegetative structures we call that is in gametophytic stage. Okay, gametophytic stage. Here now you see this one. The spores, when the spores are comes to outside in this area, after the completing the fertilizations, here formations, that is in zygote. That zygote is modified, I say, one vegetative structures we call that is in gametophyte. So here this one, this is of in gametophyte. Okay, so now what happened, this gametophyte, okay, is produced to the male reproductive organ. Similarly, second one that is in female reproductive organs. So here the male reproductive organ we call that is in antheridia, and female reproductive organ we call that is in archegonia. Okay, but how the development of an antheridia and archegonia means already you know very well. So now you see here this one, this is of in where it is in archegonia. Okay, this one here it is in young archegonia. So here it is of in mature archegonias. So this is this one total is gametophyte. Okay, sporophyte, spores, and next one this is in gametophyte. So these gametophyte are produced to the gametes. But how the development of in this gametophyte means, for example, you see here this is a gametophyte. Here one of the cell, one of the structures is going to modified as a male reproductive organ. This is a gametophyte. Okay, that's why here we are saying that is a antheridia, male reproductive organ, that is in antheridia. Okay, so one cell, two parts is there, two metatic division is going on. So how much of metatic division we are looking, that much of development. So here this of in archegonia is there. Okay, little bit flask like structures is there. Yeah, here this one, this of archegonia and antheridia. Antheridia is released to the sperms, and archegonia is a formation to the egg. After the development, anthrozoids or spores are comes to outside the antheridial chambers, and also it goes to where the female reproductive organs, where the egg is there. So when entered in the when entered the female reproductive organ, formation is a zygote. So here this after the fertilizations we call that is in oospore, zygote-like structures is there. So this zygote is modified as embryo. So after the embryo is developed as a young sporophyte. So this young sporophyte, again it is produced sporophyte. So here this is the life cycle of in Selaginella. Okay, now Selaginella, external characters and internal characters and life histories of Selaginella. Okay, yes. So after the Selaginella, yes. So after the Selaginella, so we are going to one more topic is there, that is of in equisetum plant. Okay, yeah, here I am showing the equisetum plant. This is the equisetum plant is there. Visible this plant? Hello, respond. Now you see here. So this is of in Selaginella. Here it is of in Spinapsida. Division it is a Spinapsida. Class it is of in Columapsida. Order that is of in Equistalis, and a family that is of in Equistaceae. Genus that is of in Equisetum plant. Equisetum plant. Yes. So after the Lycophyta, this is of in second one. Okay, that is of in Spinapsida classes. Here it is in Spinapsida. So this is in also sporophytic plant body. Okay, sporophytic plant body means this is also root system is there, stem is there, and leaves also we are looking in this one. Now you see here the members of equisetums. Okay, so when we are looking, yeah, before going that one, equisetum is also worldwide distributions. Okay, so almost all we are looking at entire the world. Near about represented 25 species. So most of the species were the wet places and cool temperate regions. There also we are looking in equisetum plant. So some of the species are grow where the ponds, where the lakes, where the stream banks also we are looking in equisetum plant. Okay, yes. So next now you see here. So when we are looking at this plant, it is a horse tail like structures. Horse tail like structures. Horse tail like structures. This is the plant. The species equisetum are herbaceous, evergreen, perennials, have creeping branches, rhizome from which arise erect aerial stems. These all the external features. Okay. A plant body, it is a sporophytic plant body. It is a differentiated roots and stems and leaves. Okay. Here you see this is the roots is there. The stem are two types is there. One it is in aerial branches, and second one that is in rhizome. So two types of stems is there. One it is in rhizome is there, and second one that is of in aerial stems is there. Okay. Yes. Now you see here underground rhizome. Okay. There is of in aerial erect portion is there. Rhizome and aerial shoots are having nodes and internodes. Rhizome, this is also we are looking the nodes is there in internodes, and this one also we are looking the nodes and internodes is there. Rhizome is a perennial and subterranean, growing the horizontally below the surface of the soils. Each node there is a role of scale fused at their base, into sheets. The scale leaves are without chlorophyll, means, so here we are looking two type of the aerial stems is there. One it is in one it is in fertile aerial stem. Second one, that is in sterile aerial stem is there. Okay. So what is the meaning of in fertile aerial stem? Here now you see this one, left side. So the left side, fertile shoots which contain the stobules, which contain the cones. So that is in fertiles. So which is not contain the cones, that is in non-fertile branches is this. Okay, non-branches also are looking. But now you see here, so this is of in unlimited growth, and here it is in limited growth also we are looking in this one. Okay. Yes, so next now you see here, so number of scaly leaves, various different species, and where the nodes, there is a branch of primordium alternating leaves. Such branch of primordia give rise to both subterranean as well as to aerial branches. Okay, so these aerial branches are some cylindrical, stiff, and jointed with distinct where the nodes and where the internodes. The nodes are very along, hollow, or always it is in ridge-like structures. The each ridge of in corresponds to the leaf nodes above the ridges. Successive where the internodes alternate with one another's. The internode grows by means of an, this one most important, intercalary meristems. It is situated just above the nodes. Okay. So most of the species, the aerial branches, this one, here it is in totally sterile branches. So this is all to the external features of in ecosystem plant. Okay. Yes. So next now you see we are going to the internal structures of in ecosystems. So internal, this one here it is an internal structure of the root. Yes. So here the root systems. Here the first root of the embryo. The roots are adventitious roots. Adventitious roots are cylindrical, branched, and they arise from the lower side of branches, primordia, but not directly from the node. Their origin of endogenesis of pericycles. Okay, mean these roots are coming where the pericycle is there. So generally, sir, these root internal structures differentiated three regions is there. One is in epidermis, second one that is in cortex, and third one that is in stele. Internal ecosystem root system, it is in three regions is there. One it is in epidermis, second one that is in cortex, and third one that is in stele. Sir, what is epidermis? Epidermis, it is a single layer, outermost layer. Some of the cells are epidermis bears the roots, hair-like structures, hair-like pairs-like structures is there. Okay, this one here now you see single layer, there is no any intercellular spaces, it is an outer layer, and this epidermis cell is contain some hair-like structures also here. And next one stele, next one that is of in cortex. Okay, so the cortex is so many layers is there. Outer cortex made up of in that is lignified cells, while the inner cortex it is in parenchyma. Now you see here this one, this is in below the epidermis, 2 or 3 layers is there. This is in outer cortex, and so here this one, above the steel. So here you are looking, that is of in inner cortex. Okay, so what is the main functions of in inner cortex and outer cortex means, so which contain the all type of food materials is there, that is in cortex. Yeah, and next now you see here, so above the steel, so here we are looking one special type of the structures is there, so that is of in endodermis is there. Endodermis, it is a two layer, number 1 and 2. You see now your upper endodermis, and here this one lower endodermis. Okay, yes. The outer layer is made up of longer cells, and inner layer it is in very smaller layers. This one outer layer it is in a very long cells is there, and inner layer it is in small cell like structures is there. Outer endodermis, and here now you see next one there is of inner endodermis is there. Yeah, here the pericycle is absent. Generally pericycle is absent. uh, and next one there is of steel. Okay, the steel contain large metaxylem at the center part. The xylem may be tetrarch, you see 1, 2, 3 tetrarch, or tetrarch 1, 2, 3, 4 tetrarch. Phloem is present in the space between xylem tracheids. So now you see here where this is in phloem here. This is of in xylem 1, 2, 3, 4 xylem is there. So beside the xylem is present to the phloems. Okay, so there is no pith in the root. So this is a section cutting of in root, and similarly stem section cutting. Now you see this one, stem section cutting of equisetum plant. Okay, same, almost all the stem section cutting, it is also internal features. Stem shows slight variation in different parts of the shoot. So here furrows and ridges is there, furrows and ridges. This one furrows and ridges is there, and epidermis is there, and next one that is of cortex is there, and the last one that is of steel also we are looking. Okay, yes. So next now you see here where the internode of the stems, the transverse section of the aerial stem through the internode, outlined due to ridges and furrows is there. The outermost layer is epidermis, which consists of thick-walled and elongated cells. It is covered by the, where it is in cuticle. You see here, this is of an epidermis. So this epidermis, here it is an outer layer, you say. This a cuticle is there, and in between of epidermis. So here we are looking one stoma. Okay, stomata structures also we are looking in this one. Okay, yes. So next and one important point is there where the internode section cutting, this epidermis is deposited in silica. So when you are touch rough like structures is there. Yeah, silica surface. Okay, surface is very rough. So based on that one, this is also one type of identification characters. These epidermis contain the stomata. Okay, yes. So but which is restricted in only furrows between the ridges. The stomata we are looking only there. Yeah, and the stomata consists of two guard cells, that is often overarching subsidiary cells. Okay, so this is often epidermis structures. A little variation is there with the nodes and internodes. Okay, yes. So next now you see the cortex. The cortex between the epidermis, two epidermis, lower and upper epidermis, the epidermis and steel, we are saying that is often cortex. So it is also high differentiated when where the ridges is there and where the furrows is there. Okay, so where the ridge in the outer cortex, sclerenchyma is present. So where the furrows, there is no sclerenchyma, but these sclerenchyma it gives to the mechanical support. Okay, and after this one, So here we are looking after this one. After this one, here now you see sclerenchyma cortex, this one. So where the ridges is there, where the ferrous, in the ferrous there is no ferrous. So one or two, one layer is there. So when you are looking the ridges, there you are looking more than 3 or 4 layers. Also you are looking the cortex. Okay. So it gives to the mechanical support. It gives to the mechanical support. Okay. Yes. So next now you see here. So below these cortex. So here we are looking one special type of the sections is there. So this is of in chlorenchyma cortex. Here now you see this one, this of in chlorenchyma cortex. So this chlorenchyma is elongated and compactly arranged like where the polychaete tissues in higher plants. Okay, green tissues are prominent where the intracellular spaces. This one here, where the intracellular spaces is there. Okay, so where the opposite of in stomata. The leaves are small and have only few chloroplasts. The assimilatory functions of photosynthesis carried by the chlorenchyma. So this is of in cortex structures. Yeah. So next now you see here, and where the endodermis is there. Here now you see this one. Okay. So this one, this of an endodermis, outer endodermis is there and inner endodermis. After the cortex, that is of stele is there. The stele of well defined ring, small vascular bundles are arranged a ring outside the pith. Now you see here this center part of the pith is there. So here this is totally, where is the one more structure is there. So this is totally steel also we are looking in this one. Okay. Steel, it is described as ectophilic siphonostele is there. Ectophilic siphonostele is there. The vascular bundles are opposite to the ridge and their number is equal to the number of leaves present at the nodes. So this is of steel structures. And now you see here this steel mean this bundle, this vascular bundles are collateral, conjoint and endork. Cambium is absent. The phloem is well developed. The xylem is poorly developed, as is character of in hydrophytic characters. And similarly, the xylem, it is a V-shaped structures, and here it is in endork structures is there. The metaxylem consists of in scalariform tracheids. Okay, the protoxylem tracheids become the disorganized from the cavity carinal canal is there. Okay, so these disorganized protoxylem consist of few tracheids which lie on the side of the carinal canals. The phloem is present in between the limbs. It's a V-shaped xylem and consists of phloem parenchyma, and also we are looking the sieve tubes. Okay, so this is the steel arrangement of in ecosystem plant, and you see when we are looking the stem ecosystem, some of the xerophytic characters also. Okay, so here you see one point, which one, that is of in the xylem is poorly developed. Okay, xylem is poorly developed, that is in hydrophytic characters. So now you see here, what is the xerophytic characters? That is of in the presence of in sunken stomata. Here this one, sunken stomata means, so when you are looking in epidermis, here, this area here, we are looking to the epidermis. Below the epidermis structure, that is of sunken stomata. So this is of in xerophytic characters, and also that is of in the epidermis is contain silica. Okay, epidermis contains, and this is also one xerophytic characters. Okay, and also now you see here so many air filled vallicular canals is there, reduced to the xylem, reduced to the xylem are the hydrophytic features. So this is of in unique of in equisetum stem, equisetum stems. Okay, yes. So next now you see here similarly a life cycle of in life history of in equisetums means this one. This is of in equisetum strobilus is there. Okay, this is of in sporangium structures is there. Okay, this is of in gametophytic stage is there. So before going that one, here this is of equisetum plant. Okay, so this equisetum plant, it is a sporophytic plant. Here this one cone structures is there, strobulus. Okay, so what type of structures is there means, so when we are looking the equisetum, it is umbrella shaped structures is there, umbrella shaped structures is there. Okay, so umbrella shaped structures is there. The spore producing organs. The mature plant of equisetum is a sporophyte. It bears the spores and reproduce by asexual method. These spores are producing organs of equisetum or compact structures and called this is in strobulus. This one here it is in strobulus. Okay, yes. So generally the strobulus, this one. So generally the strobulus or cones or equisetums are developed where the terminal part on the ordinary green shoot, this one, that's why we are saying here it is in fertile shoot, fertile branches. Okay, in some species they occur at the apex of short-lived fertile shoots each. Okay, the each strobulus is composed, composed of a axis from which arise roll stalked of, that is of in plate-like sporangiophores. So number of sporangiospores in each roll varies from few to many. The members of successive rolls of sporangiospores are alternating. Immediately below the rolls of sporangiospores, the axis bearing the small or large ring-like outer growth is called of annulus. So each sporangiums, this one here it is in. Okay, so here one of the structures, one of the cell is going to modified as a spores or as a spores like structures. The cone axil produce of conical. Okay, so we call this stuff in sporangiospores. The sporangium initial divides of periclinally, producing the intercell and outer cell. For example, this is the plant. So these tip of the branches, one cell is going to modified as a male reproductive organ. So first this cell is divided in two parts, inner cell and outer cells. Inner cell is attached to where the sporophytic plant body. Outer cell is going to metatic division. How much of metatic division is going on? That much of development we are looking. So after the metatic divisions, so now you see here, well, looking to the spores, this one. Okay, so here it is enough in strobulus. So after the development, these spores are comes to outside in this area, So when it comes to outside in this area, okay, So it is produced, it is germinated to, so prothallus, new type of structures we call that is in gametophytic structures. So now you see here I am showing the equisetum sporophyte plant body. What happened here? The equisetum sporophyte plant body is first produced to the strobulus, cones-like structures. So these strobulus, next one that is of sporangiospores. So these sporangiospores are produced to the sporangia. Okay. So the sporangia is contain the spores. Okay. When spores comes to outside in this area, what is going on here? It is taking food material from the environment, and these spores are germinated one new structures that is in prothallus. So we call that is in gametophyte. Okay. Yes. So now you said this is of in prothallus is gametophyte. From here to here, we call that is in sporophytic generations. So after this sporophytic generation is produced to male and female gametophyte. This one, see now here it is in Archegonia, and here it is in Antheridia. Antheridia, it is a male reproductive organ, and female reproductive organ, that is in Archegonia. Okay. Yes, so Antheridia. But how the development of Antheridia, how the development of in Archegonia means, almost all the, so one section is going to divided one parts, two equal parts. Okay. So lower part and upper part is going to metatic divisions. Yeah, here this one is Antheridia. After the development, this Antheridia, okay, Antheridia is produced to, just a minute. Antheridia, and second is of Archegonia. Here now you see this one. This Antheridia is produced to the sperms. Archegonia formation the zygote. When the sperms is going to bear the egg, here come to the fertilizations. After the fertilizations, here formation the oospores. This oospore is modified to as a embryo. This embryo is going to the new sporophytic plant body. So here this is the life histories of, this is the life cycle of in equisetum plant. Okay. So after the equisetum plant, here now you see this one, Antheridia, okay, and Archegonia, formation in this embryo. This embryo is produced a new plant. So this is the life cycle of in Antheridia and Arche. That is of in first one xylotum and lycopodium, and next one that is of in equisetums. Okay. So next now you see we are going to our, according to our syllabus, which one that is of in telome theories. Okay. Next now you see we are going to that one, our telome theories. So what is the meaning of in telome theories? Just 1 minute. Yes. So next now you see here, where is the telome theories is there? Not this one. So now I'm showing the teleome theories. So what is the meaning of teleome theories? Pterospore. Yes. So what is the meaning of teleome theories? Yes. So now you see here, after the life cycles, here the pteridophytic, all the pteridophytic plants are sporophytic plants. Okay. Yes. The success of land plants is associated with the emphasis on independent, self-sustaining sporophyte with vascular systems. These are all the lycopod. These are all the from here to from the pteridophytic plants. It is a very good, excellent vascular systems is there. That's why from here to the sporophyte is going to totally independent plant. The gametophyte is a short-living plant. Bryophytic plant, sporophyte depending on the gametophyte. But what happened when you are coming here? The vascular systems are high-fi developed in this one. That's why the sporophyte is going to is independent. Okay. So now you see here, it is believed that the primitive land plant was axial in nature. Later leaves and branches evolved the modification of the stem. The discovery earliest of tracheophytes, many botanists proposed theory explaining of appearing in the leaves, branches, sporangia on the sporophyte. Of all these theories called often teleome theory and annihilation theory. Okay, teleome theory. What is the meaning of in teleome theories? Sporophytic plant body. How the development of leaves, okay, and stem, what type of modifications, you are looking in the leaves, what type of modifications you are looking in the stem. Okay, all these development we are looking in the sporophytic plant body. So studying of all these things, we call that is in telome theory. Is it clear? Is it clear the telome theory? Please respond. Responded. Okay, we are going to detail about in this one. Now you see here. Here, the telome theory first proposed by Stephen Gimmerians. Gimmerian in 1930, there is often postulated that all the vascular plants are evolved from simple leafless and dichotomously branched axis-like of rhinia. Okay, this is the first point. You see now here, after the rhinia, you see dichotomous of these plants were known as telomes. Okay, the internodes in rhinia, when we are looking in the rhinia plant, it is a very simple structure is there. So there is no nodes and internodes. There is no small small structures or appendages like structures is there. So when you are coming in after this plant, after these plants, so here we are looking some points like structures is there. Okay, which one that is of in nodes is there and internodes. Okay, below the telomes were is called of mesomes. During the development of leaves, there was a change from the dichotomous sympodial branching as a result of fusion two or more telomes with their mesomes forming in the telome truss of in that is of in sine telomes. The telome truss may be terminate in a leaf of sporangia or sterile that is in telome of in telome or phyllite. You see here, we call this is of in telome theories. So what is the okay? Yes, so first one you see here. So Rainier plant. So there is no sporophylls here. Now one more important point is there. The process that operated in the telome concept of overlapping diffusion planations and webbing. The relation of these process to the evolution of in megaphylls shown in the figures. Okay, so from when you are coming here to here, so here we are looking to which one that is of in megasporophylls. Okay, microsporophylls. After when you are coming to that one, here it is in megasporophylls is this. Okay. Next point here. Yeah, now you see here we are looking only one simple structure is this. Okay, so when you are coming in outside, when you are coming in other plants, where you are looking the sporophylls is this. So next, when you are coming in outside, microsporophylls is there and megasporophylls is there. So all together we call that is of in telome theories. Okay. So next, now you see what is telome theory means here. According to, so his name is the most important point, that is of in Jimeranian 1930. In all the vascular plants are evolved from Rainia, like a simple dichotomous branching leafless ancestor is there, Rainia plant. So the telomes are the ultimate portion of dichotomous. The fertile telomes had sporangia and their tips, and those without sporangia are sterile telomes is theirs. Okay, so according to this telome theory, okay, primitive pteridophytes are have green algae evolved from, so green algae and those divided in all directions and gave rise to erect branches. Okay, so let on the sporophytic and gametophytic generation is developed. So this is the development from Rainia to we are coming to Selaginella. So Rainia, very simple structure is this. Lycopodium, sporophyll structure is this. Okay, when you are coming in equisitums, sporophylls is there. So Selaginella, microsporophylls is microsporophylls and megasporophylls also we are looking in this one. The primitive vascular cryptograms are very simple and dichotomously branched is there. Some branches, okay, some branches of such sporophyte developed in sporangia at their tips, and also represented at the fertile telomes. The primitive, where the primitive cryptograms, okay, primitive cryptograms means some elementary processes evolved into higher cryptograms, where that is of in Lycopsida. Similarly, second one that is of in Sphenopsida, and third one that is of in Pteridophyta. Some merits and demerits is there, means from here to here, here to here. Okay, so that is of in, here we are saying that is of in telome theory, that is of in And also one more theory is said, that is of annation theory is there. Okay, so this theory to explain the origin of microsporophylls, lycopodian leaves. The microphyll originated as superficial lateral outgrowth, or that is or outgrowth or annations on the axial side. So we call this is of in telome theories. Telome theories. Okay. So next one, see stellar evolution. This is also one of the most important point is there. So already you know very well the steel. Generally here we are looking two type of the steels is there. One which is in one it is in simple leaves is there, and second one that is in simple leaves is there, and second one that is advanced siphonosteels. Okay, so already you know very well the simple leaves, that is in protosteel. Okay, what is the meaning of in protosteels? Xylem surrounded by the phloems. Okay, xylem surrounded by the phloem, that is of in protosteels. So second one that is of in siphonosteels. Siphonosteel, it is a little bit advanced structures is there, advanced steel is there, that is of in siphonosteels. Yes, so first point now you see here protosteels. Okay, I know you see here this protosteel also four types is there. Okay, so that is of in haplosteel. Next one that is of in actinosteel and plectosteel, and mixed steel is there. Stellar evolution is there. Okay, so but scientist name is that is of in Yesu in 1953 and Smith is 1955. There are two main types of steel organizing among the vascular plant, that is often protosteel and siphonosteels. Okay, xylem surrounded by the phloems, so that is often protosteels. Protosteel are four types is there. So next point that is often siphonosteels. What is the meaning of siphonosteels? Okay, so medullated protosteel are known as siphonosteels. This type has evolved from the protosteel and it is advanced over the protosteels. The siphonosteel, the vascular tissues are arranged in the form of hollow cylinders around the center pith. Protosteel, we are looking only center part, and siphonosteel, if you are looking, so just around the center, that is often siphonosteels. Okay, so the siphonosteels are coming from where? Means regarding the origin of two different types have been, okay, so propounded by the scientists. One it is in modification theory, second one that is of in invention theories. Okay, the first one that is of in intrastellar origin pith, and next one that is of in extrastellar origin pith. Two types of so two types is there, origin of species there. Yes, so two types of theories is there. One it is in modification theories. So what is modification theories? Okay, so modification theory was first proposed by, his name is of in Woodley 1991. Okay, and Boris Thompson. According to this theory, the innermost vascular tissues are transformed of metamorphosed into where the parenchyma resulting in the formation of pith. This is often modification theory. Means here innermost, where the innermost vascular tissues is transformed as a pith. This is often modification theories. And next one more point is there in this one. The tracheids and parenchyma cells. Tracheids and parenchyma cells in the central regions of the stele gives strong support for the modification theories. Where the presence tracheids is this. The tracheids parenchyma cells in the central region of the stele gives strong support to the modification theory. Such stele is called of mixed pith stele. Okay, so all the scientists consider mixed pith stele as a that is often transitional stage between two protostele and siphonostele. So this is often modification theory. Okay, and next second one, inventions. What is the invention theory in siphonosteles? It is a proposal of Jerfy. Okay, according to this theory, pith originated by the invagination of cortical tissues into the central region, where the central regions of the stele through the leaf caps of branch caps during the course of evolutions. According to him, endodermis penetrated and inwards together with the cortical tissues to form inner endodermis. So there's a phyloglasms is there. So this of invention theories is there. So generally, how many types of siphonosteles? So these siphonosteles are two types is there. One, it is of an ectophilic siphonostele is there, and second there is of an amphiphilic siphonostele is there. So what is of ectophilic siphonosteles and what is the meaning of an amphiphilic siphonostele is there? So in this one, first one, ectophilic siphonostele means pith is surrounded by xylem. Pith is surrounded by xylem cylinder, which in turn is surrounded by phloem cylinders. So for example, this is in equisetum plant. So we call this as an ectophilic. One structure is there, second structure is there, third, fourth, five. All the structures we are looking like this, we call that is of an ectophilic siphonosteles. So what is the meaning of in that is of an amphiphilic siphonosteles? Amphiphilic siphonosteles means what is the amphiphilic siphonostele means? The xylem cylinder surrounded by xylem cylinder is surrounded by external phloem cylinder as well as internal phloem cylinder is known as amphiphilic siphonosteles. So for example, this is in Marsilia and other one that is in Adiantum. Both ectophilic and amphiphilic siphonosteles may occur in same plant in the form of continuous cylinder of vascular tissues. In this case, leaf tracts do not cause any leaf gaps, that is vascular cylinder remain uni uninterrupted. Such siphonostele is called of in cladosyphonic siphonosteles. Some members that is of in Phelicophyta and other member that is of in siphonostele be performed by the leaf cap. Such siphonostele is called of in phellocyphonic siphonostele. So this is the variation after the so this is of in theories of in so stellar evolution in pteridophytes. So one it is in protostele, second there is of in siphonosteles. Okay, so next one you see here, so modifications of siphonosteles. Appearance of the leaf caps in the siphonostele leads to number of modifications, that is of in eustele, dictyostele, polycyclic, amphiphilic solonosteles, and ectophilic solonosteles, and ectophilic, amphiphilic. Okay, so this is all the types of Siphonosteels. Yeah. Yes. So once again, evolution of stellar theory. According to his name is of Jerfi, the simple and the phylogenetically most of primitive type steel is a protosteel. In the course of evolution, the simple vascular system will undergoes several progressive changes leading the formation of complex tissues. Okay, so evolution. So this is often protosteel, and second one that is often actinosteels. So all the syllabus is totally is completed. One more topic is there. So that is often where the seed habit is there. The development of two different spores, differing size and structures, functions. The same plant, one it is in microspores, second one that is in male, second one microspores which develop into male gametophyte, and megaspores which develops into the female gametophyte is known as heterospory. So what is the meaning of an heterospores? Means microspores and megaspores. Microspores male gametophyte, megaspores it is often female gametophyte. The homospores genera sex determination is observed in where the gametophytic stage, while in heterospores genera it is observed in the sporophytic stage. Okay, the heterospory plant becomes free to grow in varied environmental conditions instead of only aquatic surroundings. It is said that heterospory is originated due to the reduction of spores number in some sporangia. The remaining spore, the sporangia increased in with the size because of the availability of more nutrients. Origin of heterospory was well observed even from ancient days. Okay, so this is of in heterospory. Okay, yes. So next now you see here. One it is of in fossil, not fossil.